Okay, so welcome everyone. So let me just admit someone else as well. Okay. Welcome to today's session. So we are going to continue into week 2. And for this week, things are getting hot quite a bit. So we are going to be looking at the TheiaProk workflow for genomic characterization. And the goals of this week is to understand the basics of the TheiaProk workflow, to be able to assess the quality of the whole genome sequencing read and assembly data, as well as understand the genotyping modules within TheiaProk. So those are 3 main goals for this week. The hope is by now, or let's say before Thursday, please ensure that you're able to run TheiaProk on your workspace, because we'll be looking at the results that we get from that analysis. So ideally, thinking about the steps for genomic analysis, you've uploaded data onto your Terra workspace. And now the next stage is to undertake these 3 things. So we run TheiaProk, we are going to get an assembly, we are going to do some QC, as well as characterize the data. So, and at the end, we should be able to interpret the characteristics of the samples that pass QC. So this is sort of like a shortened summary of the TheiaProk workflow, with the different components of the workflow here. So we'll go through the different steps, and we'll get to understand what each step does. So first off, we start with what we call read screening. And what read screening does, or rather read screening occurs at these 2 stages. We have raw read screening as well as clean read screening. And the read screening assesses the quantity of the data, and this enables the workflow to stop further processing of samples if there is insufficient data, and prevent usage of or wastage of compute resources. So think of it like the workflow is able to assess whether there's enough data for your sample, so that if it's not sufficient to be able to generate an assembly, then there is no point of moving further with that data. So at this point, the quality we are looking at is the quantity of data, not just the quality of it. And always this is undertaken at the first step of the workflow, and also repeated after the read cleaning step, which we'll talk about on the next couple of slides. So just to bring you up to speed, if you've ever interacted with the FASTQ file, which is typically the input for a lot of bioinformatics analysis, you'll recognize that we have these different lines. So we have the identifier line that identifies that particular read, and then we have the specific sequence, and then we have the plus sign, and then we have the quality scores that for each of the nucleotide base on the sequence line. And this is a standardized format that is used for all FASTQ files. So as we said, read screening checks for the quantity. So what we mean by quantity, it's looking at the total number of reads for that specific sample, the proportion of base pairs reads in the forward and the reverse read files. The reason, if I might just explain why we are looking for these numbers. So in the workflow, we have the genome length of the smallest known bacteria, correct? So we expect that any sample should be able to at least assemble the whole genome length of that smallest known bacterial sample. Therefore, if your pathogen is not even able to assemble that known smallest bacterial genome, then there's no point of proceeding forward, because it means most likely it won't assemble anything else. So at that point, the workflow stops and conserve the resources. So we look for the total number of reads, we look for the proportion of base pairs in the forward and the reverse. Of course, in this case, we are looking at Illumina paired end reads. And ideally, what we want to look for is lack of bias. So obviously, you don't want 80% of your reads, let's say in the forward read, and only 20 in the reverse. So you want a more balance. So typically, the cutoff that is used is not less than 40% on either read file. And also the total number of base pairs, again, going back to that genome length, and also related to the coverage. So estimated genome size, and also the estimated genome coverage. So all these are related, again, looking at a cutoff or threshold by looking at the smallest known bacterial genome. So we never went into this when we launched TheiaProk, but I did show you that there are optional inputs whenever you run a workflow. And if you take a look at the documentation for TheiaProk, which I posted last time, but I'm happy to put here again. So I'll just copy it and paste it in the chat again. So on that document, you'll be able to see the definitions of all the optional inputs, what the default values are, etc. So for example, here, you can see that, the minimum genome size is 100,000 bases. And therefore, that is used to estimate, like, they look at the number of reads in your files, and for, let's say, a given 10x coverage is the amount of reads that you have able to cover that genome 10 times. And of course, this is related also to, like, the minimum number of base pairs that is able to cover that genome size, and for that minimum coverage. So again, these are optional inputs, but something that we insist, like, they are not cast in stone, which means you can change, but obviously, you should be aware that when you change these things, what do you expect of the output? So the read screening outputs typically are pass or fail. And remember, we say there's a read screening step before and after the read cleaning. So if the output is a fail in the first or the raw read screen, then the workflow does not proceed. And in that case, you will not get a clean read screen. But it's possible for the raw read screen to pass and then fail after the read cleaning step. So, but ideally you want a pass for both. So this is a typical, or just a screenshot of what you'd expect to see for the read screening steps. So as an example, you'll see this first sample here. The read screen row failed, and therefore the workflow did not proceed. And therefore, you don't even have any output on the read screening after the cleaning step. For this second sample here, you'll see that it passed the raw read screen step, but then failed on the clean read screening, and therefore the workflow did not proceed beyond that. But the rest of the samples proceeded beyond those two stages of the workflow. Again, if there is any urgent question, feel free to ask, even though most of the discussion we'll have on Thursday, but if there's something pressing, please do just let me know. Yes, Rotimi.
Thank you so much, James. Um, I've gone past the use of SRE fudge. I had two failures, and then I ran the analysis for the two alone, and then I have all 42 now in my workspace. However, when I decided to run TheiaProk, I realized I had two failures again. So I ran the failures. I got failures again. I did the second time, a third time, a fourth time, and I quit. So I was wondering, is there a way I could tell why I have a failure? What sort of failure do I have? And like you explained, does it have anything to do with the quality of the reads? Maybe they don't meet the minimum threshold set in TheiaProk. I'm not sure.
Yes, so we'll discuss this on Thursdays, but yes, the sample set that I gave you for the 42 samples, I actually expect that some samples will fail. And I did that strategically so that, we can be able to troubleshoot or investigate why they did fail. So that is expected. Yeah.
Thank you.
All right. Okay, so once we've done the read screening, at least for the raw read screen, we want to go through the read cleaning step. So which occurs at this point here. And what we do at the read cleaning step is to obtain clean reads that are of high quality and without adapter sequences. As you know, as part of sequencing, usually we'll add during your library prep sequencing adapters, and these have to be removed after the sequencing. So the read cleaning step does that. And as usual, as I mentioned, of course, you have for each sequence base, you have the associated quality. And what we also want to talk about here is the Phred score, which is the probability that a base is called incorrectly, as is shown with this formula here. So a Phred score of 10 implies that one in 10 is probably called incorrectly, and this increases as you go even up to a Phred score of 50, which is one base in 100,000 bases is probably called incorrectly. Remember that this is a probability value. It's not saying that every one base after every 100,000 is wrong, which sometimes people interpret it to mean, but it's a probability value. So again, remember, this is paired end reads, and so you have forward and reverse, and it's typically known that, the reverse reads are usually of lower quality compared to the forward reads. And also lastly, for whichever read, whether it's the forward or the reverse, it's usually known that the tail end of those reads also usually have lower quality. Again, just based on the physics of the machines and how they work, it's typically expected for these machines. So with this in mind, what read cleaning step, as we said, is to trim bases that have low quality and also to remove adapter sequences. So we're going to remove low quality reads. We're going to remove low quality ends of reads as well as remove sequencing adapters. So you might have your input as such. So on the left are the forward reads and on the right are the reverse reads. And eventually you end up with reads such as this. For a given sample, you'll notice some have disappeared. That simply means that as you are trimming reads, you have a cutoff for the mean average score for that whole read. So for certain reads, you might keep on trimming until it reaches a point where the length of the read now falls below your expected read length. So let's say you expect a Phred score of 30 and above, but as you continue trimming the read, and maybe let's say your minimum read length is 70, you're getting to 69 or 70, but the read is still not having a mean Fred quality score of 30 and above. So typically, this is what you would expect of your reads after the read cleaning step. And as with the read screening optional inputs, you also have a couple of options. For example, which tool do you want to do your trimming? By default, you have Trimomatic, but if you prefer FastP, that is also an option. And also, for example, what is the minimum read length? So again, going through that documentation, as you look through TheiaProk, you'll know what the default values are. And if you want to change them, then you know what kind of values to change. So the clean reads will be of high quality and no adapter sequences. And this is what we'll use for downstream characterization. Again, remember garbage in, garbage out. So you always want to get good quality characterization. You don't want to pass low quality reads. That will end up in low quality genomes, and of course, might also impact the quality of your genomic characterization. So the data columns that you will see the values here are the read one clean and the read two clean. So these will be typically just files that have been cleaned of these low quality bases, as well as removing low quality reads, as well as adapter sequences. So just as a reminder, there is a read screening after the read cleaning step. So again, just checking if there is sufficient amount of data to proceed with the workflow. And then we have the read QC step, which occurs at this point. So what does this do? So typically the main aim of this is to identify sequencing errors. Remember, for the last two, we were checking the quantity of data, and we're also looking at the quality of the base calls. But here we are also interested in identifying sequencing errors. And here we typically use FastQ scan as a tool for this particular step of the workflow. Again, you remember we have raw reads as well as the clean reads. Looking at the number of raw reads, both in the forward and reverse, and also combined, and the same for the clean reads, forward, reverse, as well as combined. And what the CG pipeline does, looks at also the mean read length for your forward, reverse, as well as the combined reads for both the raw as well as the clean reads. So we are looking at the read length as well as the mean quality score. Remember, this is after you have gone through the read screening, the last read screening step. Okay, so we also look at the estimated genome coverage. So the tool is able to estimate your estimated genome coverage, again, based on the number of bases from your read files. And we have an optional task that is called MIDAS. Some of you might be aware of this. So it's the metagenomic interspecies diversity analysis system. And what this does is to estimate the bacterial species abundance and strain level genomic variation, or rather the contamination within non-target genus. So you have a database of more than 5,000 species. And what this aims to do is to check the abundance of different bacterial species within your reads. And if the non-target genus is identified at more than 1%, then it's advised that the QC will fail because there is you having contamination within your data set. So this is a typical QC outputs on Terra, looking at the combined mean Q value for your data, which is the clean and the raw. Obviously, the ones that, you use this for different reasons. I mean, the raw ones, it's helpful for assessing the quality of your data coming in, and the clean ones are meant to help you decide whether, these are actual reads that you want to proceed with forward downstream in your analysis. So again, lots of values here to look at, which we'll look at further Thursday. So how do we interpret these read quality outputs? So again, remember the aim of this step is to identify sequencing errors. So one of the things we look at is the number of reads. If you have very few reads, it probably means that you multiplexed a lot, and therefore you ended up not having enough reads per sample. And on the opposite side, on the flip side, if you have a very high number of reads per sample, then it means that you didn't multiplex enough, and you might need to probably downsample. There is a tool that is another workflow called Rasusa that you can use to downsample your reads, and that would mean that, which again, as you expect, most of this will be duplicates, so they add no value. Essentially, you're just making your assembly process longer. So you can downsample and then run TheiaProk. The average read quality, again, if it's too low, then most likely there's some error with your library prep. And then it means that downstream, the data is not accurate. Of course, if it's very high, that's a good thing and something that should keep up. It means that you're doing well with your library prep. The average read length, if it's too low, it means that either your library is too fragmented. Again, this might be because depending on what you're using, like did you share too much? Do you need to use probably a different kit, or just a different way of preparing your library that doesn't end up with your template being too fragmented. And then the estimated genome coverage. So again, this is per base coverage. Again, too low, probably multiplex too much, and too high, probably multiplex only few samples, and then you end up with very high coverage. High coverage is good, but too high again is not helpful, because again, it means that you could process more samples on the same run, rather running too few, and that's probably cost you more money. And as I mentioned, MIDAS will identify secondary genus abundance in your data set, and this will let you know whether your sample is contaminated with a different genus than you might have expected. And by default, this is 1% practice in the field. So if you need to change this, of course, depending on if, for example, you are analyzing data set where you expect co-infections, then you will probably expect that the secondary genus abundance will be more than likely maybe 1%. Again, this is an optional task, so you have to enable it during the workflow, and we'll see how to do that Thursday. The next step is the assembly. Again, if anybody has any questions, just let me know. So we have a set of clean reads. We've done our QC check. We are satisfied that our library prep was probably good. Everything went well, and now we want to do the de novo assembly. So that occurs at this step. And this constructs an estimation of the original genome from whole genome sequence data. And as you know, we start with an original genome, which is the sample that you collect from the patient. You extract the DNA, and then you do your library prep, which is going to be fragmented. You do your sequencing, and then you get your sequencing reads. And then after the sequencing reads, you want to go back to the original genome. And of course, you're seeing that, my genome assembly here is broken. It's not as complete as the original one. And the method that is used for de novo assembly is based on the De Bruijn graph algorithm, which we'll just look at this point. And essentially, this generates contigs from overlapping k-mass sequences. So these are sequences of length k, depending on what the tool specifically chops your reads into. So this is an example of 7 mass. So these are bits and pieces of these reads that are 7 bases in length. So you can see, it's having a sliding window of 1 base. So you have the first 7 bases and then slides to 1 window and, continues that way. So you have all these 7 mass sequences spread out across the read. And then, of course, you have many reads with this k-mass, and you use the De Bruijn graph to build or try to reassemble from these small short or rather short fragments of sequences. You're trying to reassemble the genome. And you have, these edges and you have these nodes, and this is where sequences overlap. And it looks quite straightforward. You'd expect that, when you do that, you end up with a complete genome, but typically that's not what happens. You end up with lots of knots, and sometimes resolving, this complete graph is not quite as easy. And typically what happens is you don't end up with one contiguous sequence, but rather you end up with a broken genome, or rather contigs. The assemblers that are available for TheiaProk are 4. So there's Skiza, there's Spades, there's Megahit, there's Velvet, but the default one is Skiza. And here you'll also see additional optional inputs that you can change depending on what your needs are. But typically, most of these have been optimized through several analyses, but it's good to be aware of what the defaults are. For example, what do you want to set as your minimum contig length? So you might want to vary that based on, like, well, I do have, like, let's say, a plasmid that is part of your bacterial pathogen, and maybe it's less than 200 base pairs in length, but you don't want to miss it as you're doing your assembly. So things like that will have you make informed decision on how you want to change any of these optional inputs. So for the output, we expect a FASTA file that is assembled set of contigs. So we have the column called assembly_FASTA. So this will have the assemblies for each of the samples that we submit through TheiaProk. Now, I want to talk about an optional task called assembly. No, before that, let's just go through assembly QC. I think I was getting ahead of myself. So you now have your assembly. How do you QC the assembly that you've gotten? So we have different tools for undertaking quality control of your assemblies. Again, you'll notice that TheiaProk is quite heavy on QC because, again, the workflow is used a lot for outbreak investigation. They just want to ensure that the data that you use for eventual taxonomic assignment or genomic characterization is quite good. So, as I mentioned, the tools that are used here include Quast, Basco. We also have Mama ANI for assessing the quality, of the reads here. So Quast can look at the number of contigs. So in this example here, we have 7 contigs, 1, 2, 3, 4, 5, 6, 7. And the numbers in those boxes represent their lengths. So for a good assembly, you want fewer contigs. So fewer is better. It means that you have you have less of a truncated a genome. So the second thing is the genome length, or the total length of the contigs. Again, you want this to be as close as possible to your expected genome length for that specific pathogen or taxon that you're working with. So if your genome length is 500 bases, 400 is probably a good length for the contigs, but you don't want something that's quite low. And then we have the N50 value. So the N50 value is the length of the shortest contig needed to make 50% of the genome length. So if your genome length, in this case, let's say it's 400, half of that is 200. And if you arrange these contigs from the longest to the shortest, it means that your N50 is this contig that is 60 bases in length. So the bigger the N50 value, the better, because it shows it gives you an idea of the shortest contig needed to make 50% of the total genome length. And we also have the GC content. As you know, different pathogens have their different GC content. So this is the proportion of the reads that are G and C. In this case, we have 20 bases here, and G's and C's are 9, so representing 45%. And ideally, the GC content of your assembly or contigs should be as close as possible for what you expect for your specific taxon. And of course, different bacterial species have different GC content. So you should know what it is for your specific pathogens. The data set I did give you was Vibrio, and for Vibrio, we expect the GC content to be about 47.5%. And then we have the BASCO tool, which the long form means benchmarking universal single copy orthologs. And this assesses the presence of genes that are expected to be found once in every genome, so single copy orthologs. And the BASCO database must match the genome taxa for the results to be valid. And therefore, here we have a sample of the BASCO output. You'll see that we have C and then a value S, value D values. And C looks for the completeness or the proportion of the genes that are considered complete when their length are within 2 standard deviations of the BASCO group mean length. So we can see that 99.1% here of the genes are considered complete, which is pretty good. And then out of those, you'll see we have a bracket here, S and D. So S are those that are single copy, so genes that are complete and have only 1 copy, and 0.2% are genes that are complete and have more than 1 copy. And outside the genes that are complete, we have F, so these are the ones that are fragmented or rather partially recovered. And then we have M, the ones that are missing, so 0.9% here. And then N gives you the total number of genes that were examined for that specific sample. Again, we'll go through each of these in more detail when we actually now look at the table Terra and look at each sample and just assess the quality. We have a tool called Gambit, which the long form is Genomic Approximation Method for Bacterial Identification and Tracking. And this is for taxonomic identification. So what Gambit does is, it takes in your assembly, queries it against the Gambit database that has more than 4,000 genomes. And based on what is called the Gambit distance, it's able to calculate that against a known reference from this database. It should be given should be giving you the predicted taxon from your assembly. And the other optional tool is the MAMA ANI, which looks at the percent identity or the nucleotide identity using MAMA. And this is an optional task, so it's not launched by default. You have to activate it. As I mentioned, I can show you how to do that on Thursday. And this has been implemented for PulseNet pathogens only. So those who are familiar with PulseNet, you know what PulseNet pathogens are. For example, Vibrio, E. coli, Shigella, Salmonella, Listeria, and Campi. So again, this also has a reference database. So your genome is aligned against this reference database, and then assessed against the reference that gets the highest nucleotide identity. In this case, it's Salmonella. So for this assembly quality control outputs, we expect to look at these outputs: number of contigs, the assembly length, the N50, the GC percentage, the BASCO results, the Gambit predicted taxon, and if you activated it, the taxon that has the highest percent identity and what that percent identity is. Again, this is just looking at Terra and what those columns are. So on the QC interpretation, we have an optional step here, where in this case, you can provide your own QC metrics. So again, if you need to evaluate QC metrics or your own QC metrics on TheiaProk, you need to understand that these have to be based on reasonable biological and technical expectation for the organism and the sequencing approach that you are undertaking. So the reason we emphasize on this is these metrics will depend on the pathogen. So different pathogens, will probably have different QC thresholds. And also, we don't expect that the QC thresholds, for example, will be the same when you're looking at Illumina data vis-a-vis Oxford Nanopore data. Again, QC threshold. So what do this mean? So unfortunately, comprehensive QC thresholds have not been defined for most pathogens. Some have been. For example, PulseNet has a few QC thresholds for the pathogens that they look at. And the public health community is working on most QC guidance. But then some will have to be self-defined or agreed in your lab. But then thresholds can also be wrong sometimes. So in other words, it depends. But then just that reminder that, these have to be based on reasonable biological understanding as well as technical expectations for that pathogen, as well as for the sequencing technique or technology that you used. So typically what you give the QC check module is a table that looks like this. So you have the taxon, and then you have this different quality threshold, like the number of reads minimum that you want for the raw as well as the clean, the minimum coverage that you want your reads to have for that genome, etc., etc. So it will just be an Excel TSV file that has the list of the taxons as well as these different cutoffs for the different thresholds. Okay. And what the QC check module does is to give you a pass. So it tells you that your sample has passed the QC threshold that you've given, or it can give you an alert telling you that, well, there are these things, or these metrics that your sample was not able to pass. Again, QC check does not compare all the QC metrics to thresholds, so you have to, again, spend a bit of time to manually check some of these additional columns. All right, so obviously after you've looked at your QC metrics, typically you'd want to look at any outliers in your data set. Like, are they like having very low, let's say, for example, percent identity to your pathogen of interest? Are they having very low genome length compared to the rest? So generally just looking at outliers in your data set helps you further QC your samples. And as I mentioned before, samples that do not pass QC should not be used for downstream analysis. Again, garbage in, garbage out. You don't want to end up with low quality characterization. Okay, so I know this is a lot because I'm trying to put in, compress a lot of material that typically runs for 1.5 hours within a single hour. But of course, on Thursday, we'll get more time to go through this. And as always, I'll put the slides on the training resources page. So please take some time to just have a look through the slides and watch the recording as well, so that if there are any issues, then we can address them. So we have what we call the 4 C's of quality control. One is completeness. So the question we are asking here is, do you have the whole genome represented in the sequence data and the assembly that you have? So here, the metrics that we're looking at is the assembly length, which is the sum of all the contigs for a single assembly. And also BASCO completeness. Remember that this is the number of genes that are identified to be complete. So you also want this to be as high as possible. The second C is contiguity. So how broken is your genome? So here, this is not just the length. You might have an assembly that is quite close as what you expect, but then, there are so many contigs, it's quite broken. And the metrics that you're looking at here is the number of contigs, which should be as few as possible. Looking at the N50, should be as high as possible. And also the fragmentation score. You remember we had the C and the F. So this is the number of genes that are identified to be fragmented. So this should also be as low as possible. The third C here is correctness. So if, it's as complete as you can get, it's less fragmented as you can get, then we now assess the correctness. Is the assembly correct on a per base basis, and the reads are correctly assembled? So here we want to look at the average read quality, again, going back to the Fred quality score. So typically you want this above 30s, and the higher the better. And also how high is the nucleotide identity to your target taxon? So is that value 85? Is it 90? Is it 95? So obviously, the higher percent identity to your target taxon, the better. And also your estimated genome coverage. So the average read depth throughout the genome is also a good indicator of how correct it is, because the more bases that cover each position, it means that, whatever you picked at that point as the consensus, you have a higher degree of confidence in what was called at that position. And the last C here is contamination. So are there too many reads from non-target taxa? So we say that Gambit will give you the predicted taxon. So again, this should be as high as possible. So we highest Gambit distance. And the MIDAS, which looks at the secondary genus abundance. So is that more than 1%? Is it 5%? Or is it, just way below 1%? So, and also the duplication. So again, looking at the number of genes that are complete, but then you remember those were split into those that are single copy as well as those that were duplicated, which is also an indicator of termination. And finally, the assembly length. So sum of all the contigs. So the assembly length should also give you an idea of whether, it's your target organism, because you know what you expect in terms of the length for that pathogen. So either being too long, either might mean that, your pathogen suddenly acquired some plasmid that you don't know about and it's pretty big, or, you just assembled a different pathogen that is really not what you're targeting, or it's too short, which if it's nothing to do with completeness of your genome, then it could also be an indicator that you're basically working with a contaminant that is not your target pathogen that you're analyzing. So how do we go about fixing these QC failures? So you don't have the complete assembly. In this case, you might want to sequence a bit deeper. So you have more reads that can be able to assemble the complete genome. If you're having contiguity issues, so you have fragmented assembly, then you might want to use longer read length. So probably do a hybrid assembly if you have both short reads as well as long reads, for example, from Oxford Nanopore. If you're having per base or generally assemblies correctness issues, then probably you might want to either use higher accuracy sequencing or just, again, sequence deeper, downsample your reads. But if you're having contamination, then probably you might have to just isolate and sequence your samples again, because that might be quite difficult to solve at this stage bioinformatically. So in the last 10 minutes, assuming we've gotten to the reads are clean, we've done our assembly, we've QC'd that genome, we've even optionally, taken our samples through additional QCs, we've certified everything is as we expect. Now the next step is to undertake this genomic characterization, which again is dependent on the pathogen. So remember that Gambit does the taxonomic identification, and then with regard to that, you have different characterization steps, which will be on this left side as well as on the right side. So the ones on the left are pathogen agnostic, which means they are not specific to a given pathogen. But the ones on the right, we have for each pathogen the tool that is used to do characterization. So the default generalized characterizations tasks, and then we also have optional ones. So the optional ones, the user has to give an input, and then we also have taxa specific, as I've mentioned, and these are based on that Gambit predicted taxon. So for example, the MLST, we have AMR resistance identification. So these are not necessarily based on the pathogen, but just based on the characteristics of the assembly that you get. We have plasmid identification from rep genes. Again, here is the tool that does that plasmid finder. We have AMR Finder Plus, but you also have the option of, so REST Finder is an optional tool that you can activate if you wish within TheiaProk, but AMR Finder Plus is the default one. And then we also have annotation. So you have your assembly annotated and literally ready to even submit to GenBank using Proca, or you can also activate Bacta if you wish. So again, these are the characterization steps that are not pathogen dependent. But then for serotypes, this will be taxa specific. So whether, identifying the serotype or genotype of your specific data. So how do we characterize these genomes? Of course, we have the genotype as the phenotype, and the genotype indicating, the genome characteristics used for taxonomic subtyping. As these are, for example, MLST, clade identification, clusters, lineages, as well as other forms of genome annotation. On the other hand, you have the phenotype, which is the predicted association of the genotype with a lab-derived or clinical phenotype. And this is, for example, resistance to a particular antimicrobial, the serotype of that particular sample of bacteria, as well as the predicted virulence of that particular pathogen. So we have different methods for identifying these genotypes that are based on the sequence and those that are based on part of the sequence. So like single nucleotide polymorphism, insertions and deletions, and also multiple nucleotide stretches. So for sequence identification, you can either have presence absence of a gene or may include identification of a specific allele. But for the other arm here, you're also looking at for presence absence of a mutation, and also might include identification of a specific nucleotide. So the variables that we use for genotype identification, again, for the sequence identification, we use reads and assemblies against the database, and often different thresholds for the coverage as well as percent identity. For the short stretches of sequences or these mutations, we look at the read alignment parameters against a reference genome, and then we typically have a threshold for this single nucleotide polymorphism frequencies. So percent identity, this is the proportional basis that are identical between your sequence and the sequences in the database. Percent coverage, the proportion of your sequence that aligns to the sequence in the database. We have depth of coverage, the number of reads that are marked to a specific genome position, and we also have the SNP frequency, so the proportion of reads that contain the SNP or the single nucleotide polymorphism that you've identified. There can be multiple tools for a given typing task that employ, a different method. So these tools will obviously have different pros, cons, and levels of accuracy. And obviously what you want is concordance or agreement with the different approaches that you're using. Again, remember, you might have serotyped your sample in the lab, so, on the wet lab bench. And then, of course, you're doing this also bioinformatically, so you're doing some serotype prediction as well. Obviously, if they agree, the better. If there are disagreements, then you might need to investigate why that is. It's the same thing like occasionally you will have two tools. As I mentioned in some instances here, you have MR Finder Plus or you have Res Finder, to do the same thing, but, obviously you have better certainty where they give you the same set of results. But then you might want to investigate further if there's disagreement in the outputs that you get. So just so characteristic validation, so genotyping accuracy is measured relative to a gold standard genotyping method, which is often the Sanger sequencing. and the sensitivity and specificity of the phenotypes are measured relative to the club or clinical phenotype. So obviously here it's always expected that your clinical phenotype that you identify in the lab is always the gold standard. Again, remember that prediction is unlikely to be 100% sensitive or specific. All right, so what we've covered is TheiaProk performs this bacterial de novo assembly, QC, and sample characterization. You've seen that there's read screening that allows the workflow to stop if there's insufficient amount of data. And then we have quality control steps that ensure that the data is good enough to produce high quality genomes, where we assess the completeness, continuity, correctness, and contamination of the samples. This is the end. So I realize that's a lot, but this is aimed to just give you an overview of the workflow. And then I expect that, maybe on Thursday we'll have lots of questions that we can go over. And the first question I can see in the chat is, can we start the workflow from the assembled contents? Not specifically for so if I just go back to my screen here and exit this. So again, if you go to the link that I showed you for TheiaProk workflow series. So again, you'll remember that, I mentioned, we have different TheiaProk workflows. So the one that we launched for as part of this training is the TheiaProk Illumina paired end for paired end reads. But we also do have TheiaProk faster. So this is if you have assemblies that you've assembled elsewhere and you want just to undertake the genomic characterization, then yes, you can be able to use TheiaProk faster to start from this step essentially to do these characterizations instead of the previous stages where you are doing QC and the assembly. Yes, Rotimi.
Thank you, James, and well done. And just to remind you to upload the slides, I realized that for the last training, and the slide didn't come in quite early. Maybe it came in today. I don't know when it came in eventually. I'm talking for the day, week 1, day 2. This is a week 2, day 1, right?
Yeah, so for week 1, day 2, I didn't put up the slides because essentially they were just part of the slides that we used on Tuesday. But
okay. Did so for the
yes.
Zoom session too. You didn't put that up too for
no.
for
So that one
week 1.
Okay, yeah. So that one I did put up yesterday. But I'll try to have.
Thank you.
Yeah, but I'll try to have the recording hopefully today from Kevin. So I'll ask Kevin to send me the recording. But obviously the slides, you should be able to have them as soon as in the next 10 minutes. So you'll see them in week 2 here. And then I'll just update the recording as soon as I receive that from Kevin.
Thank you very much, James.
So again, just try and launch TheiaProk. Hopefully you've been able to upload your data. You should have 42 samples. If you're having a problem uploading the 42 samples, we had, let's say, what we call a cheat sheet. So here you'll see again on the resources page, this section here, that if you are unable to manually upload these sequences, just download this TSV file and then upload it. And once you upload this TSV file, for this one, you don't even need to run SRFH because it links the file as well, but you can run SRFH if you just need to, run it and get practice with that. And then you should be able to run TheiaProk after that. Yes, Alam.
Thanks, James. So it's a follow-up question on my earlier question about using the context. So if we will be doing that, we'll need to upload the context, right? So in the first
Correct.
step, when we uploaded the reads last time, we'll have to upload the context.
Correct.
Do we follow the same steps like the manual upload, but then instead of uploading the reads, we upload the context?
Yes, so instead of this time uploading FASTQ files, you just upload .faster files. Yes,
Okay.
correct.
Okay.
Yeah, same
Okay,
process. Yeah. So if you have some extra time in your hands, you can try a few, let's say even 5 assemblies. Again, you just go to import data, go to your data uploader, you can create a new, you'll have to create a new collection. In this case, you can call them your assemblies. Again, go through the same process where you pick those files, have them there, prepare your TSV file. In this case, you might want to prepare different, have them as a different data table name just to separate them. For example, you can call them trading data assemblies or something like that. And then, once you upload them, but in this case, as I mentioned, because these is a different workflow, you'll need to import the TheiaProk faster. So I can show you real quick. We have 4 minutes over time, but if you really want to do this, then you'll click here on the Find a Workflow. Here on the Doc Store, you'll see this link here. It will open a new tab. You can search for the name of the workflow here, but that is typically very long, and the search the search criteria for Doc Store is not the best. So typically for me, what I do is I just go here to organizations. And under organizations, I look for Theiagen. And then it will give me a list of collections here. The one that we use is the PHB. So, and you'll see now we have a long list of workflows that Theiagen has developed. So it's quite long. So what I do here is just use on your browser, if you're on Windows, you'll do Control F. And on Mac, I use Command F. So on the Command F, you'll see a search box pop up. And so for me, I'm going to look for TheiaProk and underscore faster. So you'll see that, I come to that file here. So this link here, I click on it. And you can see it's TheiaProk underscore faster underscore PHB. It's not loading as fast as. Okay, fine. So once the page loads, on your right here, you'll see launch with. And because we are working on Terra, I'll pick Terra. And then it will ask you where you want to import this workflow to. So I'll pick my workspace, the demo workspace. So for you, you should be able to see if you only have access to one workspace, that is the only workspace you'll find under this drop down. But for me, you see, I have access to lots of workspaces. So I've selected my training demo workspace there. And then it will take a few seconds to import it into my workspace. And once it's done, typically it even opens it ready to just launch. So as before, you'll probably, it's still loading it. Just pick the latest version, and then if you have we can go through this if you need specific help, but at least now that shows you how you can import that workflow. I'll just select the latest version, and it's again the typical, run where you'll have to select your data table, select the samples, put in the required inputs, and then just launch the workflow. All right. it's 7 minutes past the hour. So thank you all. Please come on Thursday with as many questions as you possibly can get, so that we go through this workflow, which I hope will be quite helpful to a lot of the characterizations that you typically undertake in your analysis. So at this point, I'll just stop the sharing as well as stop the recording, and hope to see you on Thursday.